it's the just everyone talking.
Can I just check that everyone remotely can hear me?
Yes.
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 Hi, yes.
Yeah.
I don't think you can hear me. I know what I've done wrong. Just bear with me. Okay, I think everyone can hear me now.
I can. Yep.
Amazing, thank you.
Yeah.
So thank you everyone for joining. I'm gonna crack on. So today is the Optune multi-professional caregiver and patient update. Just to let you know, it is a hybrid event, so we do have people in the room as well. If I can ask, if you are remote, to keep your microphones on mute, but you can keep your cameras on. So for all of you who don't know, my name's Lily Shahabi, and I'm the neuro-oncology clinical practitioner working at Imperial, and I work alongside. Pardon? Then you can't see me. Can you hear me now? Yes, sir. Okay. Sorry. That's Dr. Williams for you. So we've got Dr. Williams here. So me and Dr. Williams are running the day. For those of you that are in the room, I'm not expecting a fire alarm. However, if there is a fire alarm, we'll have a gentleman coming in with a yellow coat and telling us how to get out of here, but the doors are just out there. For those in the room as well, the ladies are just outside of these front doors, and then to the right, it's the men's. And again, we will ask you to complete an evaluation form at the end of the day, and I would encourage you to please do so that we can continue to improve how this day is run and what you would feel you would benefit from in the future sessions. This event will be recorded so that we can upload it later onto our website, but it won't be available immediately because I will need to do a bit of editing, and that can take a bit of time. And as I mentioned earlier, if you are remote, please keep yourself on mute, but feel free to keep your cameras on. And there will be time at the end of each presentation for questions, but we will also do a Q&A at the end of the day. And you know, I don't know if any of you have ever used Mentimeter before, but if I can encourage you to scan either the QR code at the bottom right-hand side or go to this link, which Dr. Williams is going to put into the chat as well for you right now. So you can click on it. So if you've got a phone, please scan it, because I want to, the first question you will see is about what do you want? Let me. I'm going to see if technology works. So it's one thing that you would like to take away from today. And so when you click on it, the screen you see now is what you should be seeing, and you can put up to 200 characters. So yes, I can. This one. I'll give you a couple of minutes to do that. And just to let you know, I will be omitting anyone who does join in later, so feel free to join in. It will be the same Teams link throughout the whole of the day. So if you want to like pop away and come back, just leave it on and that's fine, or you can log out and come back in. And then on this Mentimeter, the first slide, you'll see this. The second is going to ask you one thing that you would like to take away from today. And then if you go to the next slide after that, it will ask you, you've got an opportunity to submit some questions so that we can ask at the end of the day. But this will be available for you to do in your own time. And then at the end of the day, we'll ask you to say one thing that you've taken away from the day from this. I am. And here is the agenda for the day. It's also available on the Eventbrite page, but for those in the room, it's all over the tables. So following me will be having Ola will talk about TTF mechanisms and basic science, and then Dr. Williams, and then we'll do a short break, and then we'll bring back the slide so you know what is going on. So without any further ado, I'm going to ask Ola to come up. And I'll allow him to introduce himself as well. But we haven't to say your surname.
It's fine.
So just the camera's up and
Well, right, okay.
that's not. I thought that was my phone. That's not. That's my phone.
That's the mic. The camera's here. Good morning, everybody. Can everybody hear me okay?
Yes.
And
Yes.
can people online hear me okay as well? Yeah, good. I heard a yes there. So thank you for the opportunity to speak today. Thank you to Matt and Lily for organising this event, and thank you all for coming. My name's Ola Rominiyi. I'm a clinical lecturer based in neurosurgery in Sheffield, which basically means I spend sort of half of my time doing research. Quite a lot of that is lab-based, and the other half of my time working clinically in neurosurgery, including looking after people that have some of these brain tumours. And so I'm going to talk a little bit today about, am I too tall?
Yeah,
Yeah. If you
Sorry.
can avoid the bald patches as well as I'm losing a bit of hair, but yeah, thank you. Yeah, no, I'm really happy to talk about some of the ways in which we understand Tumour Treating Fields works. My presentation won't by any stretch sort of take up the whole of the time, and so what I'm hoping is that if there's questions, any questions that people have, I'm happy for people to stop me during the talk if that's what people want to do. I'm also happy to take sort of questions towards the end of the presentation as well. So let's see if these slides flick forward. Do I press on here? Yeah, fantastic. So greetings from Sheffield. These are a few of our buildings in the university and in the hospital in Sheffield. And so I think the first thing that's important for me to say is that in Sheffield, we've got a really great team of people working in the tumour treating field space. And at the moment, this is primarily laboratory-based research, but we are really excited about getting the opportunity to start treating some of our patients with tumour treating fields, probably starting from next year as well. And so I work very closely with a cell biologist called Spencer Collis, and a large part of our research is tumour treating fields focused. We currently have a postdoctoral researcher in the lab, and so some of the things that I show from our lab is work that Callum's done. And we were blessed to have a previous PhD student called Aurelie, who you can see on the bottom right of the slide there, who did some of our early tumour treating fields laboratory-based work before taking up her postdoctoral position in the University of Pennsylvania. In terms of disclaimers as well, what I should say is that some of the research that we do in the laboratory is funded by Novacure. And so Callum Jones that you see on there has been supported for a couple of years via a AACR Novacure research grant, and will be supported for another few years as well. And that collaboration with Novacure also gives us the opportunity to get some of the equipment that we use to deliver tumour treating fields to cells in the laboratory, which I'll come on to talk about. So what is tumour treating fields? So we know this is a non-invasive therapeutic modality, so it's something that can be delivered through the skin. It's a local regional way of delivering treatment, so to a specific body area, not as precise as radiotherapy or radiosurgery, but certainly much more directed than giving a drug that someone takes orally, for example. And these are low intensity electrical fields, so of a relatively low voltage. And these are intermediate frequency fields. So these are, we know that if you alternate an electric field, positive, negative, negative, positive, a lot faster than this, you start to cause vibration effects of all of the molecules in cells, and that causes lots of heating. We know if you deliver alternating electric fields much slower than this, that can give other biological effects to cells. So, for example, it can start to trigger neurons to fire action potentials, to fire electrical signals along them. And for a long time, that sort of what we call the intermediate frequency range, that range of frequencies in the middle, people thought didn't really have any significant or meaningful biological effects. But actually, through a series of research looking at the effects of those alternating electric fields at those intermediate frequencies, people started to find that actually there were some effects that they had. And I think some of the earliest effects they found were the effects on how cells divide and how those alternating electric fields can impair or interfere with the way cells divide. But actually, there's a lot more to this story than that, and there's a lot more effects that those intermediate frequency alternating electric fields can have. And so when I talk to people, certainly in Sheffield and some of my colleagues that haven't come across tumor treating fields before, they feel it almost seems, quite space age. And, how can these alternating electric fields, work and have an impact on cancers? But before we even start to think about the mechanisms, I think the really important thing about this story is that clearly these fields do have an effect, and clearly they do have a beneficial effect on cancer cells. And I know Matt will come on to give a really good overview of that. But there is very good clinical trial evidence for a number of indications now that tumor treating fields can have a beneficial effect and improve outcomes and survival for patients. And so many of you will know it's approved for high-grade gliomas. It's fantastic to see some people, on treatment today in the audience, and there's been approvals for other types of cancer as well. And this is a modality which has been approved for in a number of different nations, around the world, and many of us are hoping that in the future it might be something that's approved by NICE in the UK as well. So what is the mechanism of tumor treating fields? How does it actually have an impact on cells? And so thinking about this at sort of the real base level, if you have an alternating electric field where you've got positive on one side, negative on the other side, and you're alternating, changing that field hundreds of thousands of times a second. In the case of treatment for glioblastoma, 200,000 times a second. It stands to reason that any molecule, any protein, anything within a cell that has a charge or has the potential to develop a charge will be impacted by a force, will be attracted to the positive or attracted to the negative. But bear in mind that polarity of the field is switching many, many times a second. And so any process within a cell that requires charged molecules or molecules that can develop a charge to be in a specific place in a specific time can be affected by that changing of the alternating field. And so you can imagine just thinking about the way that works, that it has the potential to affect lots and lots of processes inside cells. And I will come on to talk about some of those processes. But I remember, I was in the early days, in the days when I first started to get involved in tumor treating fields research. I remember going to one of the sort of laboratory-based meetings in the States, and there was lots of really heated and vigorous debate about well, how do tumor treating fields actually work? What do they do? Because one person says they do one thing, another person says they do another thing, and if they seem to do everything, actually we don't understand how they're working, and and maybe they do nothing, and and I found some of those debates sort of quite interesting. But I think certainly my perspective on a lot of that take was that we were expecting to understand how it works in the same way that we would expect to understand how a targeted drug works, which, you know, goes after a very specific target in a cell, and that might cause lots of other things to happen. But there's always sort of, you know, one thing, one target that's the initial effect, and everything else happens after that, or is downstream from that. You'll have to forgive my analogy, but part of the way I think about tumor treating fields working is, you know, it's almost as if you took a car and you just crashed it into a wall, you know, and the car stopped working. And then you ask, well, you know, why is my car not driving? What is the mechanism of my car not driving? It might be because the engine stopped working. It might be because you have a hole in the petrol tank and all of the petrol's completely gone. It might be because all four wheels of the car have fallen off. So there's a whole different range of ways or mechanisms it could have stopped the car from going. But actually, the underlying physical fact is that, you know, you've crashed that car, and actually it could be a combination of all of those things, which I think is probably, you know, to some extent, you know, what we see in tumor treating fields as well. You know, this is it's a physical, a biophysical way of treating cells. It has lots of different effects. If you ask, well, what is responsible for killing, you know, my cancer cells? It's really difficult to say, because it's probably a combination of all those things. And all of us in the field will argue about what we think is, you know, the most important contribution. And most of us probably think it's the thing that we work on which makes the most important contribution. But actually, there's a range of things that happen. And so the probably the earliest discovered effects of tumor treating fields and probably one of the most important effects are these effects on cell division, what we call mitosis. So how cells divide. And I will come on to talk about this in a bit more detail on one of the next slides. But essentially, when you have these molecules within the cell that need to come together and make a spindle to separate the genetic material equally into two daughter cells, those molecules have the ability to develop a charge. And so when you apply an alternating electric field, the position of those cells in the cell can be affected. So they're not in the right place at the right time, and that can affect cell division in a number of different ways. Some of the effects that we're really interested in Sheffield are these effects on DNA repair within the cell. And so there's been lots of studies, lots of work shown that when you apply tumor treating fields to cells, you get effects on DNA repair, but it doesn't affect all of the DNA repair processes. It seems to affect a very specific, you know, fingerprint of DNA repair processes. And so certainly we see these, what we call the BRCA genes, which, you know, people will know are notorious in breast cancer. We see a really big, you know, and consistent reduction in those BRCA genes. And those BRCA genes are important because they're involved in whenever you get a break through DNA, so a break through both of the strands of the DNA. BRCA is one of those genes, or one of those proteins, one of those molecules that's involved in helping repair those breaks in the double strand of the DNA. So you can imagine that if you use tumor treating fields and you reduce some of those really important DNA repair proteins, you get an increase in DNA damage in those cells. But it also has other effects on DNA and how DNA is replicated, is reproduced as well. So if you want to produce, you know, two daughter cells for the single cell, that DNA has to be doubled, and that process of replication we think is affected by how tumor treating fields works as well. A number of cells, and particularly cancer cells, have used this process called autophagy, which is essentially cells consuming parts of the cell or eating parts of themselves. And the effects of that are almost like a bit of a double-edged sword, because that process can be helpful for cells to be able to recycle and reuse components within the cells. But also that process can be harmful, because actually if cells are, you know, gobbling up parts of themselves that they need, then that could be damaging and cause cell death as well. And so what we do know is that when we apply tumor treating fields to cells, that causes an increase in that autophagy process, an increase in those cells consuming parts of themselves. There's quite conflicting evidence and studies as to whether that helps treat the cancer, or whether actually that increase in that autophagy cells gobbling themselves helps the cancer cells to be more resistant to tumor treating fields. And so at the moment, the jury's still out as to whether or not that's a beneficial effect or a damaging effect, but we know that it definitely happens. Some of the really interesting and really exciting effects of tumor treating fields are the effects on the immune system. And certainly there's a range of studies in the laboratory that show that tumor treating fields can have an effects on a number of different cell types within the body. So cells called macrophages, which can be activated to directly fight cancers and to take up and engulf parts of cancers. But also effects on cells like T cells, which help coordinate the rest of the immune system to attack the cancer. And some types of those T cells can attack the cancer as well. And the interesting and exciting aspect of the effects of tumor treating fields on the immune system, part that I find quite interesting is that obviously because the immune system, is throughout the whole body, it does raise the potential of being able to treat a tumor in one location. And actually, if tumor treating fields, sometimes alongside other drugs, can help activate the immune system to better recognize those cancers and to kill those cancer cells, it can have effects, on the tumor where you're treating. But also there's the potential for it to have some effect on tumors outside of that treatment area as well. So one of the really interesting laboratory-based studies was where they had animal models, I think they were rabbits, where they had tumors in the tummy, and they treated the tumors in the tummy with tumor treating fields. But this was a model that we know the tumors in the tummy also spread to the lungs, and these animals often get lots of lung tumors. And they found that by treating with tumor treating fields in the tummy, actually they could reduce the number of tumors that they saw on the lungs in some of those animals as well. As to how well that translates to treating in humans, it's difficult to say. But I think the potential, certainly from the laboratory-based research, is there, and it will be really interesting to see, what are the results of some of these bigger trials when they start combining tumor treating fields with immunotherapies as well. So tumor treating fields can also have an effect on the way cells migrate. And particularly, with tumors of the brain, primary tumors of the brain, like glioblastoma, we know these are really invasive tumors. And part of the reason why they're difficult to treat is because they spread and infiltrate, into the brain tissue rather than just pushing the brain tissue out of the way. One of the things we've seen from studies in the laboratory is that tumor treating fields can reduce the production of some of these things that cells use to cleave their way through that matrix of the brain surrounding them. And so there's good evidence that tumor treating fields stops or reduces the spread of some of those tumors and could potentially make those tumors less invasive. And then from a broader perspective, more recent studies have shown that tumor treating fields can have an effect on the permeability of cells, so the ability of cells to let other molecules such as small drugs into the cell. And those effects are quite interesting, firstly because the typical frequency that we find has a strong effect is slightly different to the frequency that we use clinically. So I think in some of the studies, the effects on cells and the effects on the blood-brain barrier, which I'll come to talk about, the best effect is seen at 100 kilohertz, which is lower than the sort of 200 kilohertz we tend to use when we're treating brain tumors, for example. And so, there's the potential that actually using tumor treating fields, it could be used in a way to open up those pores in cells to increase the drug delivery into those cells and to increase the effectiveness of that treatment. And because you're delivering this treatment locally, so just to the place that you really want to treat rather than the whole body, you would expect that you wouldn't get an increase in drug in other places that might cause side effects, such as the bone marrow. So that picture's gone a bit funny. But just to say that in addition to those effects that we see in sort of the cell membrane, so the boundaries of individual cells, also that protective layer within the brain, that what we call the blood-brain barrier, which separates the blood from the brain tissue. Tumor treating fields has a similar effect in opening up bigger holes within that blood-brain barrier. So it could potentially specifically increase and improve drug delivery to the brain. And what that picture should show is on one side, you have sort of a layer of cells, a sheet of green, which are some of the cells that normally make up that blood-brain barrier, and there's no holes in it. And then after treating with tumor treating fields for 48 hours, what they see is a really big sort of pores, openings within that barrier, which you could see quite clearly drugs would be able to get through. But the nice thing about that effect is that it doesn't seem to be a permanent effect. So it seems to be that you treat with tumor treating fields, those pores open, and then when you stop within 24 hours, actually all of those holes seem to close. And so you can see there's a whole range of effects that tumor treating fields have. Certainly we could debate about what people think is the most important. I would have my views, lots of people would have their views. In Sheffield, we do lots of research specifically on the effects that tumor treating fields has on DNA repair, which I'll talk about a little bit later in my presentation. But just to say, we're really interested and excited in those potential effects that it has on DNA repair. Because at the moment, the main treatments that we use, particularly for brain tumours, are DNA damaging treatments such as chemotherapy and radiotherapy. And so for those cancerous cells to be able to survive, when you damage the DNA with those treatments, they have to repair that DNA. And so if you can interfere with the way those cancer cells repair the DNA, then potentially you could make chemotherapy and radiotherapy a lot more effective. And or you could reduce the dose that you need of chemotherapy and radiotherapy to have the same effect. But just to give a little bit more detail on those effects of tumor treating fields on cell division. So we talked about a little bit about the way cells divide, and they have to form that spindle, what we call a mitotic spindle, in the middle of the cell, where the genetic material is all lined up in the middle, and then those. Is that sidebar in the way, Lily? How do I move it out the way? Can I minimize that? Is that causing a problem to anyone online? No. Good. I'm just going to get a quick drink of water. Yeah, so for the cells to be able to divide, they have to form the spindle in the middle where all of the genetic material lines up in the middle of the cell. And then you have sort of this web that equally divides that genetic material half to each side to form two daughter cells. The way that spindle forms is that you have these small molecules that we call tubulin. You can see that sort of those two little balls at the bottom. And these are able to become charged. So all of the electrons within that molecule can move to one side, so that one side is more negative and one side is more positive. And to form that spindle, thousands or probably millions of these little tubulin molecules need to join up together, so that you've got sort of a long ropes of those tubulins. And so that process requires those molecules to be in specific places at specific times. But actually what happens is, in the context of that rapidly alternating electric field, these molecules just can't get to where they need to be. And so it's more difficult for that spindle to form. And then it's more difficult for that spindle to accurately pull that genetic material apart and separate the genetic material equally into both of those cells. Later on in that process of cell division, where you have these two cells which are now sort of starting to separate away from each other, and so you've gone from having sort of a very round, football type shell to almost like an hourglass figure with this, band in between those cells. There are other molecules which are involved in helping those two cells separate, and they have the potential to develop a charge as well. And so as tumor treating fields interferes with the movement of those cells, actually the cells can find it more difficult to separate out between those from one cell into two cell. And actually quite often you can see sort of like a persisting bridge between those two cells, which shouldn't be there because they're just struggling to sort of separate. And I think I've hopefully got some good videos of that a bit later, because I've not pressed on there. And so just to say, really, there are places within that process of division where when the cell is round, that electric field is fairly uniform across the cell. But as those cells start to divide or pull apart from each other and you get that narrowing in between them, what they find is because of the way the field impacts that irregular shape, you get much higher field strengths towards the middle of the hourglass structure. And so that higher field strength in the middle is sort of pulling those charged molecules back towards the middle of the cell. So the cells are trying to separate away from each other. And actually the physical effects and forces that electric field is applying to those molecules is almost sort of like trying to pull them back together. So it's like this epic tug of war between these two daughter cells that are trying to separate out, and the effects of the alternating fields, which are pulling those molecules back towards each other. And so you can imagine that causes lots of issues with how the cells divide, whether or not they can divide, how that genetic material is separated between those two cells, whether it's separated equally or unequally. And actually quite often cells can just die during that process of trying to divide because it's become so traumatic. And I think these are both showing at the same time. Let me just stop that. Wrong. So hopefully what you can see on this left-hand side, I'll just go back to the beginning, is a video of I would say a normal cell, a cancerous cell dividing without any tumor treating fields being applied to it. So you can see the cell rounds up, that genetic material, which is in red, sort of makes that spindle and then splits into two daughter cells. And the first thing that I should say is that all of these videos that I'll show are sort of played at the same speed. So I'll just show that again. You'll see a sort of a single cell. It rounds up into a ball. You can almost for a moment see a little spindle in the middle, and then it separates out into two quite quickly. Let's just show that again. So that process is relatively quick, relatively efficient, and perhaps too efficient, because that's why these cells, these cancers are so aggressive and continue dividing and dividing. And now the next thing I wanted to show is a video of the same sort of cancer cells. So these are not brain tumor cells. These are HeLa cells, which I think from a cervical cancer, and just see how that process differs. So we can see the cell, we can see the genetic information in red. It forms that spindle, it tries to divide, but you've just got this persistent bridge between those two cells. It's almost like they're sort of struggling to divide. And I do have a few more videos, but I think the way this is coming up, it's not going to let me show the videos behind it, but I'll just show that video again. You can see that cell, it rounds up. It's trying to divide, but it's almost as if it's being pulled back together, and that process of those two cells separating in two is just a lot more difficult. And I don't know if you can make it out, but what you can also see is there's a little bit of genetic material here, which should be in the nucleus, or in sort of, with all of the rest of the genetic information in that cell. But actually that's just out there by itself. So even when genetic information, is taken into the cell that it should be, and actually you might get what we call micronuclei, small bits of genetic information which are where they shouldn't be, and that can cause problems. That could also activate the immune system to help sort of target and mop up these cells because there's something that's not quite right with them. I'm going to see if it lets me go to the next slide. There we go. Brilliant. And so what I also wanted to show briefly is just some of the equipment that we use when we're delivering tumor treating fields in the laboratory, because you need a system to be able to treat cells so that we can do some of these experiments. And so we're fortunate to work in collaboration with Novacure, because what that gives us is access to this system, which is called the in vitro system. And this is how we deliver tumor treating fields in the lab. So I'm not sure if you can see my pointer online. Yeah, but we have this base plate, and then we have these little ceramic dishes that we can put cells in, that we put into that base plate, and these deliver that tumor treating fields. And you have sort of electric fields going across one way, and then you have electric fields going across orthogonally in the other direction, to be sort of more similar to how we treat in the clinic. And this just shows one of those base plates with those dishes in there, and we've got some cells and some media in there to help the cells grow. And then those electric fields are generated by this these generator systems, which we have these long cables which we hook up to these base plates. One of the things that we've also been working on in Sheffield is just trying to find what we think are good ways of testing tumor treating fields in the lab, because whenever we're testing something in the lab, we want it to be as representative as what it would be like as if we were treating cells from a patient. One of the systems that we use is this scaffold based system, so that rather than growing cells on a sort of flat culture, we grow them in this it's essentially, it's like a polystyrene scaffold, and we cover it with a material that the cells like to stick to, and so the cells don't just grow on top of it in a single layer, but they grow into it as well. And that just helps the cells to grow in 3 dimensions and make connections to each other, just like they would in a normal tumor. And so those sort of sheets of that polystyrene material are in the bottom of these plates, and we can put cells in there, we can put media in there. But importantly, we can take out those scaffolds and we can put them into this tumor treating field system to deliver that treatment in the laboratory. And that's what that looks like. And then after a period of time, we can start to look at what effects that those treatments have on those cells. And so one of the ways we do that is we put a chemical on, which shows if there's any cells that are alive, they convert that chemical into these purple crystals. And then we can start to get these, what we call clonogenic results, where for every individual cell that we place into that scaffold, over a period of 2 to 3 weeks, if it grows and proliferates and is able to keep growing, it forms one of these dots that we call a colony. And we can start to count those to see how effective a treatment is. So if you've got less of those, you know your treatment is hopefully more effective and killing more of those cells. If you've got more of those, then your treatment is less effective and you're just allowing those cancer cells to grow. So working with Novacure, they've kindly sort of done some simulation of the how tumor treating fields gets into those scaffolds and how strong the intensity of the tumor treating fields is within those scaffolds. And one of the things that we found quite interesting is that when they did sort of simulations of a mouse brain, actually, because a mouse brain is very small compared to a human brain and sort of very homogeneous, very similar throughout, they found that you get a very similar intensity of the strength of that field throughout the mouse brain. So if you look at the scale bar here, the brighter that color is, the stronger the field is, and if it's darker, that field strength of the tumor treating field is less strong. And in the mouse brain, actually, what you see is that you've got pretty much the same color throughout the mouse brain. So you get strong effects on the mouse brain, and it is very similar throughout the brain. Now, obviously, we use mouse models quite a lot in laboratory-based research to try and predict what happens in human patients. But actually, when they've done those same simulations on the human brain, what you find is because of the size and the shape and the irregularity and the ridges within the brain, you get big differences in the strength of the tumor treating fields, in different regions and different parts of the brain. And so one of the things that we think is good about that scaffold model is when we run the simulations on this, because of the material it's made of, actually we don't just get sort of a similar strength of tumor treating fields throughout the scaffold, but we get pockets where that intensity of the field is less strong, and we get pockets where that intensity of the field is more strong. So in some ways, we think this is a good model, but as with all things in the lab, there's never a perfect way to try and represent a really complex human disease. So I think the next couple of slides, I just wanted to show some of the work that we've done looking at tumor treating fields and how it affects DNA repair. And going back to my comments previously about, well, if tumor treating fields if we think it affects everything, then maybe we don't really understand it, and maybe we've not really got to the root effect that it's having. And to some extent, maybe that is there is some truth to that. But certainly when we look at DNA repair, both in Sheffield, in our lab, and other groups around the world who are also looking at DNA repair, we see very similar effects, very consistent effects, and very reproducible effects. So every time we do these experiments in different types of cells, we find very similar things. So this is one group of glioblastoma cells, tumor cells, grown from the same patient. One is from the sort of the core of the tumor, that's the light blue. One is from like the invasive edge of the tumor, that's the dark blue. And we've looked at a number of these proteins which are involved in DNA repair. And what we find is that some of those proteins, like those BRCA proteins, which are involved in repairing strand breaks in DNA, very significantly reduced. So one would be sort of like the baseline, what you would expect if there was no change. And we see a big reduction in the amount of those proteins after treating with tumor treating fields. And that's in that 3D scaffold growth system that we showed earlier. If we look at another set of cell lines, so this is cell lines from a different patient, but also taking cells from the core of the tumor and that invasive edge part of the tumor, which invades into the brain. Again, some of these really important DNA repair proteins like BRCA, so that's just come up, BRCA1, BRCA2, we see a big and consistent reduction in those proteins. And when we look at work that our colleagues have done in a completely different laboratory, based in the States, it's a bit more difficult to see this, but when they've looked at, again, those BRCA proteins or some of those other proteins like what we call FA pathway proteins, they found exactly the same things. They found, so in cells which are not treated, you have, a good baseline level. And then when you treat with tumor treating fields in a whole number of different cell lines, you see a reduction in the expression of those proteins. And again, that's really important because those BRCA proteins are involved in repairing damage to DNA. So if you can stop cancer cells repairing that damage to the DNA, you can make those DNA damaging treatments potentially more effective. And so to try and prove that concept, our team has been doing work where we take drugs which we know turn off certain DNA repair processes, and seeing if we can use that to make current treatment and tumor treating fields more effective. And so this is work done by one of our previous PhD students, Aurelie, and she's looked at this drug called a PARP inhibitor, olaparib. So this is a drug now which is approved for a number of types of other cancers like breast cancer. And these drugs are really interesting because initially when they were first approved, they were approved for people that had those BRCA mutations, those breast cancer associated mutations. And part of the science behind why those drugs, worked really well in those patients is because those BRCA proteins, they are involved in repairing strand breaks in DNA where you've got a break through both of the strands of DNA. And PARP inhibitors, protein PARP is involved in repairing when you've got a break through a single strand of that DNA. And so what you have is that you have a situation where if you've got a break in single strands of your DNA or half of your DNA, that PARP protein can repair that break. But if you get rid of PARP, then lots of those single strand breaks or breaks at a single side of the DNA would start to accumulate until you get breaks on both sides of the DNA. And so normally then the cell would use a different way of repairing those breaks on both sides of the DNA. But actually, if your BRCA is not working, then you can't do that as well. So you get a single hit, you get lots of single hits, you can't repair those. That causes some double hits or some breaks on both sides of the DNA. And because of the problems with BRCA, you can't repair those as well. And the interesting thing about using tumor treating fields in a similar way is that if you can use tumor treating fields to reduce BRCA protein in cells, then you can make a tumor behave like it's a BRCA mutant tumor, which would make it more sensitive to PARP inhibitors. And that's essentially what we find. So normally PARP, if you've got cells with PARP in it, it causes these addition of these chains to protein, and you get this black smear. When you treat with a drug that turns off PARP, that smear completely disappears. So we know that the drug's working. And when we start to combine those drugs, those PARP inhibitors, with tumor treating fields and radiation, essentially the interesting thing here is all of these plates, these four here, have been treated with radiation. If you have radiation and you have that PARP inhibitor, that drug that turns off that PARP protein, you get a lot less cancer cells growing. And if you add in tumor treating fields to that combination, you get a really effective treatment. And that graph just shows a similar effect of that. And I think the important thing here is that at the moment, out of these three treatments, patients in the UK are getting radiotherapy, obviously alongside the chemotherapy, but they're just getting radiotherapy. And so if you start to think about if you add in tumor treating fields and could add in some of those targeted drugs as well, you've got the potential to really increase your potential to kill cancer cells compared to what patients get at the moment. And this bottom row just shows very similar concept. Oh, yeah, go ahead. Question at the front.
Tell me if I'm wrong on this. On that Petri dish,
Yeah.
the eight Petris, isn't there a difference between unmethylated and methylated types of tumour in terms of their ability to do what this starts talking about? So
yeah,
this irrespective of the type.
Yeah. Yeah, that's so yeah, I'll repeat the question, and it was a really good question. So the question was around looking at the results that we see with these combination treatments. If you've got a methylated tumor, so a tumor that's methylated for MGMT, so it's not expressing that protein, which is involved in repairing a certain kind of DNA damage, or you've got the other group of patients where they have unmethylated tumors. Does that have an effect? Does that have a difference on how effective that treatment strategy is? Is that the question?
Yeah, you sounded.
Yeah, so this is a really interesting area and one that we're actively looking at in the laboratory. So certainly that top combination with a PARP inhibitor, radiation and tumor treating fields, we've tested that in a number of cells which are methylated and unmethylated, and it seems to have a very similar good effects. We don't see sort of a big difference between the two. However, as you probably would expect, when we look at that combination on the bottom, so with temozolomide chemotherapy, we know that MGMT directly repairs the damage that temozolomide causes, to the DNA, and so we do find a difference between the MGMT methylated and unmethylated cell lines. So we find that if cell lines are methylated, that combination, which includes temozolomide chemotherapy, is much more effective, but we do still see a good combination effect in the unmethylated cell lines. So it's not like it doesn't work, it's just that it doesn't work as well, which you would kind of predict. But again, with the radiation, actually, it seems to work really well in both sets of cell lines from both sets of patients. So yeah. And sorry, I said I would give sort of lots of time for questions at the end. It's taken a bit longer than I would have expected, but just to say in summary, so tumor treating fields, this is a non-invasive way of delivering treatment. It uses low intensity alternating electric fields at an intermediate frequency, so not so slow that it starts to fire off action potentials or make muscles twitch, not so fast that it causes huge amounts of heating, but somewhere in the middle. That does still cause a little bit of heating, but not as much as those much higher frequencies. It's used clinically in a number of countries and reimbursed in a number of countries. Obviously, it's used to a small extent in the UK, but it's not currently approved by NICE on the basis of cost or cost effectiveness. And in terms of the way this modality works, those alternating electric fields, positive, negative, negative, positive, and switching hundreds of thousands of times a second, just impacts a force, causes a force on any molecule within a cell that either has a charge, of which there are many, or has the potential to develop a charge, and causes things to not be in the right place when they should be, and that can affect a number of different processes. And of those processes, I think probably some of the important ones are the effects on cell division, the effects on DNA repair, the effects on how cells move, and the effects on the immune system as well, but there are probably many others. And with that, I say thank you. Please.
Thank you so much for that, Ola. I'm just gonna stop sharing. Can everyone see us? Well,
Yeah.
not me and Ola. Before, does anyone in the audience that are virtual have any questions that they would like to ask Ola from his wonderful presentation? If you want to put your hands up or just unmute yourself. If not, does anyone in the room have a question?
Just like to ask if there's what the science shows about long-term effects of Optune, sort of 3-5 years, something like that. Is there a gradual improvement, or does it cease to become useful after a time?
Yeah, I mean, I think certainly in the. Yeah, no, it's a really good. Oh, sorry. So the question was about the long-term effects of Optune, and if you've been treating, for example, 3 years, 4 years, 5 years, what impact that has, on the cells and on the effectiveness of treatment. From personal experience, we've not tested those longer durations of treatment in the laboratory, because I think technically it's more difficult for us to do that. We are starting to look at sort of treating cells for slightly longer periods of time and then sending them off for profiling, where we look at all of the different genes in the cell and see what the effects of that is, over a longer period of time. But that's results that's in its early days, and I wouldn't like to say what we will find yet. I think Matt will have a good perspective on what happens sort of clinically and to patients treating for a long period of time with Optune and the effects that you see there. So sorry, I know that's not an answer really, but it's just to kind of say there is some work in this space, and hopefully maybe next year we'll have a bit more of an answer for you.
Yes. Gavin, would you like to unmute and ask your question?
Yeah, great presentation, Ola. Nice to see you. If you could choose any compound that you have tested in the laboratory, which we know how difficult it is to set up clinical studies, especially when it involves different companies. But which compound would be your choice?
That's a really good question. Obviously, I'm probably biased because I have the most knowledge in the area that we're working in, and certainly from the work that we've done so far, the best combination responses that we get are with the ATR inhibitors, so including AstraZeneca's ATR inhibitor, and so good in fact that we're looking to take that into an animal model, with some funding that starts from early next year, and we're very keen to look at the potential for combinatorial trials combining that ATR inhibitor with TT fields. So so maybe that is something that we need to talk about, and maybe that's something that we need to talk about with matters as well. Yeah.
Thank you.
Hi, Faye.
Hi, Ola. Thanks for that presentation. It's really nice and clear and comprehensive. I'm interested in your use of the patient-derived cell lines and thinking about heterogeneity. It's interesting, the comparison between cells, from the margin and from within the tumour. I just wondered if you've any insight into differential effects across cell states from sort of proliferation to quiescence, or whether you've looked at cells derived from patients up front, newly diagnosed glioblastoma, and patients with recurrence, and whether you see any differences there.
Yeah, so I mean, I guess talking not specifically, but just about our tumor treating fields work now, but in terms of our laboratory research, looking at models where we've grown cells from different parts of the tumor, we have sort of started to look at the representation of different cell states within those models. And certainly in some of those models that I showed, what we do find is we see enrichment or more of this mesenchymal state in those distant brain invasive parts of the model, which in some ways conflicts with some of the data and the work that's out there at the moment. We have theories as to what it is, but I think that's probably quite a specific discussion to have maybe outside of this forum. But yeah, happy to have a chat offline or do some email back and forth. The other thing to say is that we've got lots and lots of those models of heterogeneity where we've got cells growing from different parts of the tumor and from patients that have got disease which has come back, recurrent disease as well. And we're very happy and always keen to sort of share those resources with the research community so that we can start to get better treatments that work against all different parts of the tumor.
Thank you. One more question in the room, and then I will move over to Dr. Williams.
One remotely, which was, will Sheffield be running trials?
So just to repeat, that was Dr. Williams asked that remotely. Someone has asked, will Sheffield be running these trials?
Yeah. So that's a really good question, particularly because in Sheffield, compared to other areas in the country, we've, historically, we've not been able to offer anywhere near the amount of clinical trials that other centres have been. We're incredibly excited that we are going to be a centre within the upcoming Keynote D58 clinical trial, which is tumour treating fields combined with immunotherapy. And so we're on track at the moment to hopefully start recruiting into that trial towards the end of Q1 next year. That will only be for newly diagnosed patients, and certainly very much part of my job is looking to see what other clinical trial activity I can encourage our team in Sheffield to get involved in, so that we can have more options for patients locally in South Yorkshire.
Thank you.
Just a quick note, is that for recurrent?
So that trial is only for patients that have newly diagnosed tumours. I am at the moment having discussions with a company which makes a completely different type of treatment for recurrent glioblastoma, and a treatment that looks quite interesting, and we're very interested in it because it has an effect on DNA repair as well. But I can't confirm anything. That's early discussions, but I'm hoping that we'll have something that we can offer to people that when their tumours come back as well.
Thank you so much,
Thank
Ola.
you.
That was such
Thank
a wonderful.
Sorry,
No, we are running.
No, we were running slightly late.
Just bear with me. I'm just going to do one little test.